So hello everyone who joined today's webinar, and hello everyone who's gonna watch the recording. My name is Maryna, I'm from Cube Biotech, and I'm happy today to moderate our somehow a bit unusual Infusion Masterclass. So I'm today not alone. I have today co-speaker with me, the John Ozbal from Momentum Bio, who's gonna, be together, yes, and we're going to present some new data from our collaboration with Momentum Bio, basically. And yes, so I'm very happy to have you all here today in our Infusion Masterclasses. They are done from researchers to connect within the field, to share the new findings, to get the feedbacks. And yes, so we have something also nice to share today. So some shared projects, collaborative project of Cube Biotech and Momentum Bio. So thank you very much for being here today. Yes, so we're gonna start with the talk right now and gonna tell you something about affinity selection, mass spectrometry, high-throughput screening on the GLP1 receptor. And John gonna introduce his technology and discuss on the advantages, and we're gonna tell what we did for this part of the project. And basically after the talk we're gonna have a question and answer session together. So whenever you have any questions during the talk, you can write them down in the lowest right corner of your screen, so that we can highlight your question afterwards on the screen and then have a nice discussion afterwards. Yes, so again, thank you very much for being here. So can we have the slides on the screen, and I'll just give you the word.
Yes, thank you. Well, thank you, everybody. I appreciate the opportunity to talk to you about our research here. We've worked with Cube for a couple of years now. ASMS is a technology that has been becoming a lot more popular these days as target areas such as degraders, molecular glues, and the like have become more and more prevalent. It's a high-throughput binding assay platform, allowing us to identify small molecule ligands towards proteins and protein complexes. An area of growing interest has been membrane-bound proteins, which are inherently challenging, just both expressing them in the right structure and formation, but also because a lot of membrane-bound proteins have either lipid structures or hydrophobic domains for which you can get non-specific binding. And as a result, you get a lot of hits that aren't meaningful. And I'll discuss to you how we work with Cube to solve some of these challenges using the GLP1 receptor as a case study. A little bit history of Momentum Bio. The core team at Momentum has actually been together for almost a quarter century now. It's hard to believe. But initially, we commercialized a technology called Rapid Fire. This is a high-throughput mass spec front end that enables functional assay screening primarily, but we've now used it for all kinds of other stuff as well. If you're in the high-throughput world, you may have heard of it. This company was sold to, or this technology was sold to Agilent back in 2011. And after working there for a few years, the core team left to start a CRO, originally doing only rapid fire-based functional assay screening. But in the last decade, we've expanded our portfolio to a wide range of high-throughput screening technologies. And very excitingly, just a couple months ago, we acquired a company based in Munich, Germany, which is one of the reasons why I'm here in Germany with our Cube partners right now, called Omics Scouts. And Omics Scouts is a global leader in proteomics applications with over 20 different proteomics assays and workflows that are commercialized. This now allows us to start with screening assays, find interesting compounds, but then further characterize those in downstream proteomics applications. And I got a couple slides just to briefly go over what it is that we can do there. We have now over 34 mass specs of all shapes and sizes now across 2 different sites, one in Munich and one outside of Boston, including a wide range of triple quads and a very wide range of high-resolution mass specs, TOFs, QTOFs, Orbitrap systems, and more on order. We also have a compound library. Currently, it's 300,000 compounds of a diversity set for looking at to potentially screen for binders as ligands as various targets. And also a covalent library. A covalent irreversible screening is a big part of our business as well. And with that library, we have all the compound management services, echo, et cetera, to be able to pool and screen those. Our business is along several major verticals. The kind of more conventional, generally triple quad based small molecule business. This is both functional assays, but in general, non-regulated preclinical bioanalysis assays, PK, biomarker assays, et cetera. You can imagine all those assays. More recently, we're getting into radioligand research, looking at ICP-MS, looking for generally tumor surface antigens that we can then target with a radioactive ligand. Of course, we do studies with a non-radioactive isotope. It's actually something that we've been talking with Cube Biotech as well as to expand our collaboration. Again, these are membrane-bound cell surface antigens, so we can express and then potentially find ligands for and help clients in developing new medicines. Our binding assay platform, both covalent and non-covalent, are by far our biggest service verticals, ASMS being by far the biggest. And here we can screen literally hundreds of thousands of compounds in a matter of days, looking for small molecule ligands. Covalent binding, while not currently our biggest vertical, is our fastest growing. There's a huge amount of interest in irreversible binding assays to essentially inactivate proteins permanently. Proteomics is something we've been working on for a couple of years, but now with the acquisition of Omics Scouts has grown dramatically. We now have an entire range of both DIA and DDA, TMT or native type analyses, and I'll cover those a bit more in the next couple of slides. We can also do things like biomolecule QC and some large molecule work as well. I'm going to dive into our ASMS platform in some detail. As I said, if there's questions and so on about other screening technologies, we can always cover them at the end. All right, so there's lots of ways to do what is known as affinity selection mass spec, but they all kind of start at the same level. You mix together a target, that's typically a protein complex, or a protein oligonucleotide complex, with one or more small molecule potential ligands. You then do a rapid size-based separation, big versus small, and then you interrogate the large molecule fraction with LCMS. They can be decoupled. The first step can be done offline in a gel filter plate or something like a speech green assay, or this can be a integrated single-step process, which is what we use. It's, the system we use was originally called Alice and commercialized almost 20 years ago by a small group called Neogenesis, which was then acquired by Sharing, which is now part of Merck, and they still publish on this and work on this routinely. Here's what our system looks like at a high level. In our initial assay, in a 96 or 34 plate, we mix together our target. So this is going to be a protein, a complex, or an oligonucleotide with, in our case, 250 small molecule test compounds. These are your putative ligands. This mixture is at equilibrium when it's waiting to be analyzed, but when the analysis begins, we do a rapid size exclusion column based separation. Right? In this case, large molecules and small molecules are separated. The matrix in this column is a 3-micron bead, but they're porous, roughly 60 angstrom pores. And what happens is that the small molecules, the Lipinski's like roughly mass 320, 330 molecules, will have to diffuse through those pores and take a very long time to come out. But the large molecules are excluded from the pores, they're far too large and elute in the void volume. We take this early eluting void volume and integrate it by conventional reverse phase high pressure UHPLC-MS, ultra high pressure LCMS, and integrate by a high resolution mass spec. Now we know which compounds are in each pool. And so if we see a compound in the detector, the only way it got through the SEC column was it was bound to the protein and it got dragged through. So we have a putative binder. At this stage, we don't know how tightly or how selectively. We just know that we have something of interest because it met our criteria to be called a hit, and it's a putative binder. We can then take those hits and interrogate them in downstream assays in many different formats to both verify binding and characterize it as needed. Here's a picture of our setup. This is the first dimension, the SEC column. So this isn't some gel filter plate. This is an actual high pressure LC column. There is a high pressure switching valve right here. And the second dimension, reverse phase column, where we're going to interrogate the binders. Now, in the first step, we're trying to promote ligand formation. I want the protein small molecule ligand to survive through this first step. This actually is chilled to 4 degrees. In the second step, we actually dissociate the ligand from the protein. This is done by heat. This is heated to 60 degrees. It's high organic because it's a water signature gradient eluting off the column. And it's also very high acid because there's 0.1% formic acid in the running media. So essentially the protein precipitates out, the ligand is released, and it's detected by LC-MS. Now, between these two dimensions, you can see up top is a UV cell. So we did a UV trace, which looks like this. We have an early eluting large molecule peak. These are things that didn't fit in those pores, and a late eluting small molecule peak of things that aren't binders. Now, these all came off as a single front here because at 1 minute, we switched the SEC column from an aqueous phase to an organic wash, and it just flushes everything out through the system. And so it's this first peak that's the interesting one. Everything else actually goes to waste. I'm going to zoom in on this peak in the next slide. So this green shaded region is the region that we're actually diverting to the second dimension and interrogating by LC-MS. You'll notice that we actually purposefully let this heel go. This is where things like detergents and medium-sized molecules start eluting. You get a little bit more signal, but a lot of more background. So experimentally, kind of ignoring that trailing peak has been much more successful in our experience. And so any small molecule that happens to elute in this phase, in this peak, is by definition somehow bound or interacting with the protein. Here is an example of a hit in one of our screens. I picked a molecule that happens to have a halogen of bromide in it. Here's a structure. This was not from our particular case study, but just a random hit. What you can see on the top trace here is the UHPLC chromatogram. So we have a nice Gaussian peak eluting at roughly 3 minutes. And at the bottom, you're seeing the mass spectra. So this is the isotopes of this particular molecular formula. The way we do our hit ID is driven not by molecular weight, but by molecular formula. So it's hard to see here, but you've got red boxes. Those red boxes are the theoretical isotopes of this formula, and inside of them are black lines, which is the experimentally determined data. So 2/3 of bromine is a heavy isotope. 1.1% carbon is C13. 17 carbons, you'd expect roughly a 17.7% of the molecules to have a heavy carbon. Well, there's your C13 isotope, there's your heavy bromine isotope. Some molecules, by coincidence, have both a heavy bromine and a C13 and 2 or 3. So you have this expected isotope distribution. And one of our hit criteria is a match between the expected and observed isotope patterns. Now, one thing else I'll put out here, the area under this peak correlates to the concentration of this compound that we detect. So if you've got a very, very tight binder, the protein is 100% occupied, you'll have a large peak. And if it's a weak binder that's only 10% occupied, you'll have less of a signal. Now, between compounds, you can get very, very different mass spec ionization. So one compound at a micromole can give you 1 million counts, another might give you 10,000 counts. But for a given compound, that ionization is normalized out, and the concentration of the detector will determine the size of this peak. Now, protein occupancy is driving the peak size, which in turn is driven by how tightly the compound bound to the target, which means that you can actually infer binding affinity, at least in terms of rank order, based on your peak area. So this is ideal for things like selectivity screening. If you want to see if your target binds your on target, but not a dozen off target proteins, you can actually compare the peak areas in that experiment and get direct a rank order of the binding affinity, and therefore figure out if you've got a selective binder or not. Again, it doesn't tell you what the binding affinity is, it just allows you to rank order them. Here's another example we're talking about. On the left, you've got now a non-halogenated test compound, but we've got both a protonated and a sodiated attacluster, and the expected isotopes on the left and the theoretical on the left and the experimental on the right. Obviously, I picked examples here of very, very good matches, and this is scored from 1 to 100. Typically, anything over about a score of 75 would be considered a hit, and lower than that might be artifact or noise. Now, that is only one of the criteria we use for our hit selection. We actually have an entire custom in-house data analysis software package called Aston. We have a second analysis package that we call Dalton that's actually a compound management software. So as we create these pools of 250 compounds, our Dalton software will create a database file, which is fed into our analysis software, Aspen. In this particular case, I just picked an example, well, E12, there were 3 compounds that met our criteria for a hit. So these criteria are talked about the size of the signal and how good that isotope pattern match is. It's also how unique the signal is. One thing we do when we run our screens is we run a DMSO blank control for every column, every row of the plate. And we interrogate these 250 compounds in well E12, but also in the DMSO wells. And if we see equivalent signal or similar signal between the DMSO and the target well, clearly it's not test compound specific signal. There are no test compounds in the DMSO, and that is automatically filtered out. We have another level of uniqueness, and that's our cross-well filter. We then look at the compounds that passed in well E12 and interrogate it across the rest of the experiment. It could be several hundred injections. There could be another hit in another well that has the same molecular formula and the same retention time, but you're not going to get 50 of them. So typically, if it's more than 5 hits in different wells, we'd also call that an artifact and eliminate it. But these are all real-time selectable criteria that a user can use as needed. The way that we typically want to screen, I kind of put the campaign funnel here. First of all, on the right are our conditions. Each injection is 10 microliters. Our target concentration typically is at 5 micromolar. Now, for our GLP1 assay, we actually wanted a much lower concentration due to non-specific binding. I'll talk more about that in a bit. But our test compounds are at 1 micromolar each, and there's 250 of them in the pool. Now, this is just our default conditions. We've actually run 500 compounds at 0.5 micromolar before. It comes down to the DMSO tolerance of the assay. We'd like to say below 2.5%. So you could go to more compounds, but then you have to work with the concentrations. To put things in perspective, if you were to run a 5 micromolar, we're doing this as an excess of target such that if we have weak binders, we have excess protein to be able to see multiple hits in a well and not start to lose weak binders due to competition. But 10 microliters, 100,000 compounds would be about 400 injections. That's 4 mils. 4 mils of a 5 micromolar protein solution is 1 mg of protein for a 50 kD protein. So it's not nothing in terms of protein consumption, but compared to some other biophysical methods like SPR, it's nothing. It's really very protein friendly to screen large numbers of compounds. We almost always start a project with a pilot screen. In our case, it's a 16 pool, 4,000 compound mini screen. We're looking for hit rates, hopefully around 0.5%, but anything between 0.1% and 1% is usually quite solid. We don't want so many hits. 5% hit rate will have thousands and thousands of hits that you have to interrogate. But we also want enough hits that we can do some clustering and maybe some initial SAR as well. So that 0.5% is about right. Once we're happy with that, we can move on to our primary screen. Now, if the hit rate's typically too high, the biggest problem with protein ASMS isn't not enough hits. We almost always find hits. It's too many hits. And the reason is that you've got a sticky protein, you've got some non-specific binding, and you're finding true binders, but they're not useful compounds in the sense they're stoichiometric and selective. We can play with some detergents. We can add a little bit of 20 or something, sub-CMC though, which we can then hopefully get the compounds more soluble. We can play with concentrations of proteins and ionic strength, but there's not a lot to play with there. So assay development is usually just a few rounds of a couple of different buffers, and we can, 3 times out of 4, the assay storage buffer, the protein storage buffer actually works just great. We'll then move on. Once we're happy with that initial pilot, we can screen our full library. We'll then confirm our hits in a second round. Typically singletons are up to 5 compounds. Again, 5 micromolar protein versus 1 micromolar test compound. You can have up to 5 compounds with a competition. And confirmed hits can then be followed up in dose response to figure out the KDs, selectivity screening, competition assays, etc. So competition assays where we can put 2 compounds to see if they bind the same site by altering their concentration and seeing if we compete one off. But we can use this to our advantage in a screen. Say you have a target that is an ATP binding protein, and you're not interested in ATP competitive hits because, well, they could be much harder to make selective and make a good drug out of. Not a problem. We simply add ADP or ATP gamma sulfate or maybe a sport if it's a kinase to the buffer and essentially block that site. So we'll enrich for allosteric hits. You can do the same thing for an enzyme. You can block the active site with an inhibitor, etc. I've listed here some of the advantages of our platform. So the assay development is almost nothing compared to a functional assay where you have to worry about electromagnetic kinetics and dose responses and linear ranges and KMs, and we're just seeing if things stick, right? So the analogy I can make is we throw spaghetti at the wall and see if it sticks. You can paint the wall a different color, but it's still the same assay. It's very, very straightforward, and usually we're screening within a matter of days. We can screen about 100,000 compounds in just under 2 days. We talked about the protein usage. It's about a mg for 100K screen if you're at that 5 micromolar concentration for a 50 KD target. Fully solution phase, right? Some other assays like DEL require you to immobilize to a membrane, to a mag bead or to a surface. In this case, your protein can be completely unlabeled, no tag needed. But more importantly, your small molecule test compound doesn't need to be tagged either. Like in a DEL screen, you need a 50-mer oligo tag so you can barcode these things. No need. It's fully solution phase. There's really no limit to the size of the molecules we can work with. The example I'm going to give are membrane proteins with nanosystems, but we can work with the largest complex we've worked with to date is a 12-protein, 2 million Dalton complex. We've actually screened an intact nucleosome before. This is definitely allows us to work with a wide range of targets. Also importantly, when we do find a hit, the singletons are in the freezer. We can just go pick them as singletons and do the follow-up assays that afternoon. Something like a DEL screen requires you to actually resynthesize those hits off-label and then come up with an entire secondary assay to validate them. In our case, it's very, very rapid. And I talked about how the ASMS signal actually quantifies or correlates to the protein occupancy, target occupancy, therefore very effective for selectivity screening. I have a slide here comparing ASMS to the other very, very popular binding assay, which is DNA-encoded library screening. DNA-encoded library screening is incredibly powerful in that you can screen billions of compounds very rapidly and very, very cheaply, right? It uses next-gen sequencing on those barcodes that the test compounds are made with. Now, the way you get to those billions of compounds is usually through combinatorial chemistries or 2 or 3-step syntheses of fragments. So this does somewhat limit the chemistry. You usually have the planar chemistries, but clearly people can find lots of hits with DEL. Whereas in ASMS, we're able to screen a much wider kind of 3D structures and Lipinski's compliant, rule of 5 compliant molecules. But there's no question they can screen billions of compounds. The real advantage of ASMS is in the fact that it's a native solution phase label-free assay. And when you do find a hit, you can go follow it up immediately. You don't have to work. A lot of the work with DEL is downstream. It's post-screening, resynthesizing compounds, and coming up with secondary methods to do them. However, one thing we do a lot of is clients who do DEL screening will then send us those compounds, which they found, and synthesize off-label to verify by ASMS. So ASMS actually is a decent orthogonal tool for DEL. There's a question about our chemical library. So we have a 300,000 compound library. It's pre-pooled at 250 compounds per well and divided into 6 50K subsets. Now, this is a commercially available library. You bought it from multiple vendors and assembled it with really diversity in mind. So that was the main key driver here, when as much chemical space as possible. The pools, each pool of 250 compounds is actually mass coded. In other words, there's no 2 compounds with the same molecular formula, and they're all at least 0.2 mass units away from each other, such that when you find a hit, you're pretty sure you know what compound you have. That's not a requirement. You can actually randomly pool compounds as well. And if there's 2 compounds that are too close in molecular formula or identical molecular formulas that we can't resolve, our software simply calls them both hits. And then when we do a confirmation screen, we'll screen them separately and figure out which is the real hit. We also have a small microcycle library, an RNA-focused library, as well as a covalent library. But as I said, we are completely library agnostic. We can screen client libraries and also third-party acquired libraries. In fact, we have several large pharma libraries we store in-house of companies based in Europe, several of whom we visited just recently, because the shipment logistics of hits from across the pond can be a real limiting step. So we've pulled our library, we screen them, and we find a hit. We can go cherry pick them from our own freezers and follow them up immediately. Here's some just overview of the library itself. As I said, these are in 6 50K subsets. So this is the full library. We've really tried to make these as rule of 5 compliant as possible. And here's some of the criteria we use for those rules. There's different versions of this, so I put them up here, but generally speaking, a highly diverse library. And we can certainly provide you with more information on these if you'd like. And I will also point out that these are not singleton libraries. There are several compounds potentially around a common structure. So there's some SAR that we might get off of a hit if we're lucky, which actually did happen in our example. Once we do find a hit, we can then follow up with dose responses. So this is the same exact hardware, but instead of running pools of compounds, we now run our hit at multiple concentrations. Everything else is the same. We're hoping to get data that looks like this. So on the right, we have a very, very tight binder. It's a nanomolar binder. Actually, this is a sterosporin and a kinase, so I'm cheating a bit, but it's a very, very tight binder. And what you see here is as we titrate the test compound concentration, the signal goes up until we get to the full occupancy of the protein. Right now, there's no more room in that binding pocket for sterosporin to bind. So the excess compound is being filtered out in the SEC column. And so the signal goes up and saturates. Now, I can tell from this curve that it's a very tight binder. We reached 100% occupancy almost immediately, but I can't actually calculate the KD. I just know that it's less than 2.5 micromolar. On the left, you have another example. This actually was an mRNA target, not a protein target, but you can see it's a much weaker binder. The KD is half the hill slope, so in this case, about 6 micromolar. What you can see is that it took almost 4-fold excess of test compound to get to 100% occupancy because it's a weaker binder. But this can be curve fit, and then we can, in this case, we also ran a no target control to show that the signal we're seeing is actually target specific. Now, this is real data, but I did cherry pick the best looking real data for this presentation. But this saturating curve is what we're looking for. If we do see a curve that looks like this on the left, right, here's a curve that never saturates. So this is a real binder. It actually occupies the target, but it never saturated, indicating that it's probably non-selective. It's probably superstoichiometric. More than one test compound is binding the protein, and as a result, the signal goes up and up and up and never saturates. And here on the right, we have an example of there is a specific binding pocket, and a good one. It's going to be a low single-digit micromolar binder. But when you really drive the equilibrium towards a test compound, you drive a secondary weaker interaction, and hence the signal starts going up again. So I'd actually cut this curve off at 20 micromolar, maybe potentially rerun it and it skip the high concentrations. So it's probably a less soluble compound with some non-specific binding as well. With that, we're going to talk about our example here, but first I'm going to hand over to talk about a bit about how this protein was actually generated and how Cube Biotech works.
Yeah, thank you very much.
Yeah.
So that's really exciting to see. So this powerful technology and Alice screening with the high throughput and in comparison, low sample consumption. Yeah, so how much data you really can get out. That's incredible. So thank you very much for introducing us to this technology and for introduction of Momentum Biotechnologies. So now I'll take over and tell you a bit about about Cube. Yeah, so and how we stabilize the GLP1 receptor and what kind of the sample we deliver to the Momentum Biotechnologies to perform this collaborative project in the end. But first of all, let me introduce to you about some facts about the Cube. So basically, we are headquartered in Germany, but we have also sites at the US. And we work together with very different countries in terms of the selling our services or products. And now, after we acquired Ozbal Life Sciences, we are a team of more than 100 people helping people in their research innovators and helping to solve different problems. So in Cube Biotech, we have different paths how we can basically help you with your membrane protein research. And of course, we have on the one side, we have our product portfolio. So you can work, you not necessarily need to work with the membrane proteins. So we have also solutions for soluble proteins, but we have a strong technology for stabilization of the membrane proteins. And of course, we help out if you need a purification. So our second part is the services. So whenever you have or don't have like enough expertise to work with, or you want to outsource your project, or you have really tough target which you just cannot really crack on, so you can make use of our services. So we go all from the expression, stabilization, and purification. Can perform characterization of your target and basically also perform structural determination and go and upscale and produce the membrane protein at any scale you need. And the third part, which exists basically from the last years, we thought that it would be nice also to facilitate the research on membrane proteins in terms of reducing the risks and costs for our customers, and came up with the NativeMP targets. So we're thinking about the proteins, what might be interesting for the drug discovery purposes, and making them accessible for our customers. So they are already pre-screened, so you just basically can come to us and inquire this protein, and we can produce it to you. We see ourselves as a drug discovery enabler, so we can support your drug discovery programs from basically from the moment that the target was identified, yes, and work on all of the process. And we're trying to create a kind of a partnership, collaborative ecosystem, yeah, so where you not only getting a protein from one provider and then go to the other company to do your screen, for example, yeah. So, but this is especially the reason why we go in these collaborations, yeah. So to create this value in the end for your process, yeah. So that you come and you do everything in the one space. So every process you need in the preclinic, so we can basically support this phase with the target protein of your choice. This is a bit more deep dive for what exactly we are doing here at Cube. So we start with the expression. We select the best expression host for your target. Then the heart of our technology basically is the solubilization screen with our NativeMP platform, which consists of more than 30 different synthetic copolymers. And what we do, we end up with a protein that is stabilized with this kind of lipid copolymer complex, and which I'm going to explain you in the next slide. And basically, depending on the project, what you're doing, yeah, so we can also make an add-ons on different protein-specific characterizations. If it's just something that we always include, something like size analysis or thermal analysis, or is this page Western blot, but also some binding assays, also to prove that the target is actually active in the end. And then on demand, perform also upscale of the target. So speaking about our technology, so what makes our technology different from the classical methods, for example, for working with the membrane proteins? So obviously a membrane protein would sit in the biological membrane surrounded by the lipids. And the classical way how people do this already for a lot of years is to use a detergent which works for certain targets, but the problem that you have is that the detergent washes away almost all of the lipids that surround the protein, and you kind of miss the information, yeah, so together with these lipids. So we see that most targets are compromised in their functional stability, and that may be a high risk for the project on the long timelines. Our technology, we don't use detergents at all. So we employ synthetic copolymers, which is a part of our NativeMP platform, which basically wraps around the membrane protein. There is stamp out the membrane protein out of the membrane together with the host cell lipids, and stabilizes the protein without detergent. And we basically, because the lipids are there, yeah, so we can preserve the functional stability of the protein, and we can even perform the assays at the body temperature. Because for example, we speak about the human targets. They are we have there now a body, and it's like 37 degrees Celsius now when we speak about the membrane proteins. So this is also a bit of a sneak peek as to how the technology works in detail. So we have the synthetic copolymers that wrap around the protein, so they interact predominantly with lipids, and it's completely detergent-free. And what we get is basically your membrane protein. It's kind of a snapshot of the moment, how the protein exists in the membrane. In this way, we kind of solubilize it and stabilize it in one step. And the beauty of this technology is that you don't need to add any polymer afterwards. So once you solubilize, you can purify your protein, and you can treat your membrane protein as it would be the soluble one. And this is the beauty of the system. How is it possible that we can stabilize so many proteins, as for example from our catalog? Our NativeMP platform consists of 5 different backbones from synthetic copolymers, and in total we have 32 different synthetic copolymers that can we use in our screenings. So that's why the probability that we're gonna solubilize your target basically is very high in this moment, no? And what we also offer here at Cube Biotech, we can also not only do the stabilization and solubilization of the protein with synthetic copolymers, but we can also provide them as a functionalized versions. For example, with attached biotin or a fluorescein for detection purposes, or here for the immobilization purposes, or put any other tech of your choice and specifically designed for your application. So there is no need anymore to modify the protein itself, because our philosophy in Cube Biotech, we would like to work as native as possible, and this employs also working with wild type and full-length protein. And because the particle, so the protein, stabilized in the synthetic copolymer particle, you can treat it as a soluble protein. It also opens a door to the variety of different applications, which are sometimes difficult for the proteins that are stabilized in detergents. So you can perform screenings and validations using MSD, SPR, GCI, and so on. You can do high resolution structural determination. You can develop biologics in vivo and in vitro. And also, you can do small molecule drug development, as for example, using ASMS. So featuring already a bit sneak peek of the data that I'm going to present in a couple of minutes. Okay, this is a bit here the overview about our NativeMP targets available at Cube Biotech of off-the-shelf proteins and off-the-shelf projects. Whenever you see some familiar target on this list, so don't hesitate to reach out to our team, and we're happy to help you with the production of this protein. And as you can see, that's protein from very different classes, because there is not a specific class that NativeMP system works on, but we can basically work with every single membrane protein. And you would ask probably how it is possible that we have so many proteins on the list, and in this basically short time. We started to do this list last year. So we use our proprietary patented technology for fully automated membrane protein workflow. We developed this workflow for the solubilization and the purification, basically in fully automated manner, and which allows us to screen the targets in less than 3 hours, and we can screen basically several targets in the same day, so to say, facilitating your and making faster, so all of the membrane protein purification, solubilizations, and so on. So coming a bit closer to our case study about the GLP1 receptor. So when coming to this collaboration and showcasing, so how method for using that you use, so it's can be combined with our stabilized membrane proteins. So we're searching for a target that is kind of, everyone knows this target in the end. So if you haven't heard about the GLP1 receptor, you heard about Ozempic and Wegovy. So the two medicines that are very popular in the last 2 years, because the semaglutide, which is basically the active compound or the active component so of the system, is basically targeting the GLP1 receptor. And was made for treatment of the type 2 diabetes, and for obesity, so especially for Ozempic. So Wegovy was done, but developed for more in direction, so that's for people who wants to lose weight, for example, so but they are both of this medicaments, how you call it. Well, the drugs, in the end, they need to be taken subcutaneously, and it's always nice to have an alternative, like a small molecule, so that can be taken orally, probably also reducing the side effects, and possible. So that's why we selected especially this target to showcase how the system gonna work together with ALS screening. So this is how the GLP1 receptor looks like in our hands. So we stabilize it with NativeMP platform. So we perform the screening, and we perform the two-step purification. So basically that's always an affinity purification. And then we do the size exclusion chromatography. And you can see here nicely the band of GLP1 receptor. And this is actually the first ever full-length wild-type stabilized GLP1 receptor. So especially GPCRs often they fail when you work with a detergent, but here we have a nice sample. So what you see here in the DLS analysis, a very nice homogeneous sample. And what's important for this target, we performed some MSD measurements with the antagonist Exidin-4, and we proved actually that the protein is available to bind something. And yes, so you can see that we got AC50 values, so near to the literature values on cell-based assays, and that was basically the function of stability of the GLP1 receptor was preserved, and was the target of choice to proceed with in this collaboration project. Also, just worth to mention, in a lot of different applications, you would also need some nice control or reference sample, for when you work and screen, for example. So what we also offer at Cube Biotech, and what also we sent it to Momentum Bio, was the so-called NativeMP, NativeMP nanodisk. So basically, when we stabilize a protein, membrane protein target, we can also prepare this empty copolymer nanodisk, and we use in-house prepared lipid extracts from the culture of choice. So if you work with a heg culture, then it's going to be lipid extract from the heg culture. If it's insect cell culture, then that's going to be a lipid extract from the insect cells. Yes, and we use the same copolymer that we use for the target protein. So in this case, from the GLP1. And also the feature here, so we can also do this in biotinylated and non-biotinylated version whenever you would need, for example, to mobilize something or not. Yes, and basically these two samples, the GLP1 are stabilized in our NativeMP system, as well as empty copolymer controls. That's something that we headed over, to see how it performs in your system. So now I would like to give it again over to you.
Thank you very much. Appreciate that. Yeah. So as you can tell, we had a great protein to start with. We've worked with membrane-bound protein systems before, typically as you said, detergent solubilized systems. These are extremely challenging because the micelles that the detergents cause can be massive, right? Millions and millions of Daltons sometimes. So the load of lipid compared to the target itself is massive. You have even a tiny amount of lipid binding, which a lot of compounds do in the presence of these membranes, can lead to huge amounts of background signal. So the protein binding is essentially masked by this lipid binding. In the nanodisk world, the lipid load is much more reasonable compared to the protein itself. So things that bind the lipid even somewhat, you can actually see the protein binding signal over the lipid signal. The difference of how we ran this project from what I said before is that we actually ran an off-target screen simultaneously with the target screen. Right, so the off-target screen was nanodisc, the blank nanodiscs versus our target. Now, if you remember, I told you that we do a DMSO subtraction. We actually added one additional filter, and this is part of the workflow for membrane proteins. We interrogate the same compounds on and off target, and in this case, we use a signal factor of 2. If we had less than a 2-fold signal average for the target than the off target, it was assumed to be a non-specific binder to the lipids, and eliminated as a hit. So only compounds which selectively bind the target at a 2-fold signal window or more were actually considered. Everything else was done with our usual pathway. Here's the UV chromatogram of the target as well as the blank nanodisc. You can see significantly more signal coming from the target. We've got some other stuff coming out later in the unbound compounds, but this green shaded region again is the region of interest. You can see, we can see the target and off target. So these were run as an A/B screen in the same batch, right? We ran a row of the target containing GPCR, GLP1, as well, and then followed by the blank nanodisc, and went back and forth row by row in the same batch. Here are the criteria we used to call a hit, minimum 5 times background signal levels, et cetera, and at least a 2-fold affinity for the target over the blank nanodisc. We ran our standard 4,000 compound pilot screen, and these conditions resulted in 6 unique hits. I put their formulas and our internal compound ID numbers here. Here's an example of one of the compounds that was a hit. There's a structure, the mass spec. It's a very nice, clean looking hit. Because there were only 6 compounds, we just went straight to dose response to follow them up. Normally, if you had a large number of compounds, we do a first single point confirmation, followed up by a dose response, but with only 6, we thought it was a good idea. And you can see here a range of fairly typical results. The top right corner here, compound 518, is clearly an artifact, right? This made it through our filters, but is not any sort of binder. It's just random noise, and so that is not real. The compound on the bottom left here, compound 701, has a very weird-looking structure, curve rather, most likely a poorly soluble compound. You can see the top end of the curve is falling off. So this could be a good binder, but it's hard to draw a good solid conclusion from this particular data. It could be a non-specific that's solubility issues. In any case, we won't fall. The 2 compounds on the top here are actually very nice looking dose response curves, maybe one with some biphasic behavior. These 2 compounds we actually reran in the presence of just blank nanodiscs to ensure that we're getting signal coming from target binding and not some sort of an artifact. So you can see there was very little binding to the nanodisc in one case, some in the other, but clearly much more binding to the target itself. This gave us enough confidence to actually move into a screen. So we followed this up with one of our 50K subsets, in this case PS7. Here are the characteristics, or just the averages of the of that particular screen we ran. So a very Lipinski's rule of 5 compliant subset of our library. This was screened, again, similar conditions. We ended up with 75 hits as a result of that initial experiment after all those filters were applied. And we took those 75 hits and ran them in singleton, plus minus target, right? So nanodisk alone or target in nanodisk. And 34 of those actually ended up confirming as hits. Now, we typically shoot for a 50% confirmation rate, right? We don't want to define a hit so tightly that we get 100% confirmation rate because we're bound to miss weak hits. We also don't want to define hits so loosely that most of what we find are artifacts. That 50% rate is kind of what we're shooting for in our filters, and they were somewhat designed, the criteria I gave you earlier, to lead to that 50% rate. So it's somewhat subjective, but we're trying to get enough material that makes sense we can work with, but not so much material that we spend more time doing confirmations than we do actually screening. Here are the plate. It's 199 pools in this case. The number in each cell is how many hits we found in that well, in the primary screen and in the confirmation screen. So you can see in well B3 here, there were 4 primary hits. I mean, one of them confirmed, but essentially half the compounds confirmed. So what we're hoping to see here is some normal distribution of hits, right? If they all come from one column or something, that's not a good sign. You'd like to see some sort of a normal distribution. Now, this plate was run by row. And so you can see that we actually have a fairly decent, so this gives us confidence that we're probably finding meaningful hits. You can all see we have our dimer controls and our positive controls. In this case, we didn't have a positive control for GLP1, so it's actually a different protein positive control. It's carbohydrate hydrase and chlorothiazide. That's just a check engine light to make sure the machine's working. Those confirmed hits, as I said, they were confirmed by running on and off target. So the blank nanodisk versus the target nanodisk. So the peak area you see for the target and the off target and the ratio. We use a cutoff factor of 2 to call it a hit here. If it's less than 2, we're just assuming it's probably non-specific binding to the nanodisk. So most of them didn't bind the nanodisk at all, the blank nanodisk at all. And some of them did, but at a much lower concentration or much lower peak area. So they're considered hits. I'm going to show you an example of 6 of the compounds we chose to follow up here. So one of them ended up being the lower right one here, compound 680. It's a non-specific binder, right? It didn't curve over. But the rest of them actually are binding with nice saturating curves with some level of affinity. The top 2 here, compound 517 and 520, are actually very close structural analogs. One's a methyl, one's a chlorine, but otherwise are identical molecules. And you can see that small difference led to a 3-fold difference in affinity. So by coincidence, we also have a little bit of SAR in here. As I said, that was just, I picked these examples as well. We actually run all 34 compounds in dose response. I'm showing you some of the nice looking curves. They didn't all saturate. There was a good number of non-specific ones, but overall some very, very nice data. And with that, I'll tell you a bit about the kinds of stuff we can do next. This particular example was done as a case study to showcase working with membrane proteins. Most of the work we do, of course, are for clients specifically, so I can't talk about it. But this example we did, on our own in order to be able to present and give you a capabilities list. With our Omics Scouts team, we're now able to follow up these hits in a wide range of proteomics assays, looking at in-cell experiments, looking at protein abundance, et cetera. So if a test compound actually has a downstream function, that could be signaling, that could be stability, that could be expression. We can follow those up with proteomics experiments. We actually have a list of about 15 different scouts. I've given you a list here. I won't go through these all in detail, but essentially we can interrogate these compounds in cell-based assays in a wide range of applications. Very much, of course, target dependent. Some targets are, some assays might not be relevant for some targets, but gives us the ability to go ahead and do downstream assays. We also have some information products. Most exciting is our turnover atlas, where we have 44 different cell lines where we quantify the protein turnover rates using SciLac experiments. And so that's just available for immediate acquisition. If you're interested in a protein and like to know how long it lasts in a cell, especially for covalent studies, that data is probably already available. And with that, I'd like to conclude the presentation. Thank you for your time, and we can take some questions.
Yeah, beautiful. Thank you very much.
Thank you.
So
Thank you.
for being here, and we were so excited by the time to see this results. Yeah, so that it works so greatly. So thank you very much to being doing the part of this project and showcasing. Yeah, so that's yeah, the membrane proteins, they are not a problem anymore. So we can target them pretty nicely, no? Okay, so let's go to our question and answer session. I'm gonna read the question because maybe someone just not watching the video but hears us. So the first question is, this will only catch small molecules that are bound to the structure. What about the transient binding of small molecules?
So when the compound, the target small molecule mixture is awaiting to be analyzed as equilibrium, on rates and off rates, right? So technically everything's transient, right? You got on rate, off rate. If your on rate's much higher, you'll have a tight binder, vice versa. There is a limit to the weakest interactions we can measure here. During the SEC column, we separate the big molecules and the small molecules, and so on rate stops. For 12 seconds, you're losing ligand as it goes through the column. Now, we chill this to 4 degrees to maximize ligand formation, but still, if you've got a 2-second off rate, you're going to lose multiple half-lives and if already a weak binder. So our limit of detection for binding is typically 50 micromolar or tighter. If it's much weaker than that, the off rates generally are on the order of seconds, and we just don't have enough signal to detect. So this is not particularly a good method to find like fragment-based binding where you might have millimolar affinities. Those off rates are so fast that you need another technology like SPR or rotational NMR spectroscopy or something like that.
To cross-validate in the end, yes, for sure. Yeah. So, what are the selection principles for your 300K library? So
this was very much based on maximizing diversity. So we have 4 different vendors. Actually, our library is growing. Our next 50K subset should be ready in a few weeks. We're looking to get as much chemical space as possible. It's not optimized for kinases or some specific class. Because we work with clients, we have tons of different application areas from helicases to DNA binding, and you name it. So we try to go as diverse as possible and be kind of class agnostic.
Okay. Can you give a protein and you find the best SMA? Yes, I mean, for sure, that's how we work. Yeah, so in the end, I mean, we're not only employing the SMA for the screens. Yeah, so but as I said, SMA is only one from 5 different backbones that we utilize. And the good thing about the copolymers of the new generation is basically that they have improved physical chemical properties and improved solubilization properties. So it's even worth to take a look not only at the SMAs but also on the different backbones. But for sure, that's just contact us so we can speak about this. Do you see a false positive detection problem due to the interaction between polymer nanodies and small molecules? If so, how do you reduce the false positive detection?
I think maybe the question came before I went through the presentation, but yes, indeed, non-specific binding to the polymer itself is a huge issue, which is why we're doing the blank nanodis subtraction. If you weren't to do that, you would end up with many true hits. They're binding the construct, but they're not useful from a drug discovery standpoint. And so by doing the background subtraction of the blank nanodisk, we're able to get rid of substantially a large part of those non-specific binders.
I think that's maybe also the influence of the lipids in the system may not be underappreciated, I think, in this moment, because we're speaking not just only about the membrane protein, but also surrounding lipids.
Correct,
so, and that's
correct,
why this counter screen with the empty copolymers is contain the original lipids from the culture. Yeah? So that's a good control in this case. Yeah?
Yep.
Will it work if you have a lead compound and want to do SR? I feel like this will be a terrible since a small molecule have the same core structure.
Actually, as I showed you in the examples, we actually support clients with SAR all the time. Now, there's actually a large number of different technologies out there that you can use to look at protein binding, from SPR to mesoscale to ITC. There's other systems out there, thermal shift assays, DSF. ASMS does have a huge advantage, especially when you're working with protein-protein complexes, right? Because it can be really difficult to immobilize a complex. You typically immobilize one molecule with part of the complex and hope that the complex itself remains. ASMS is solution phase, so there's no need to. So in the case where your target's a difficult one, or your target's an oligonucleotide, which you do a lot of, we support clients with SAR routinely, and you can tell twofold and better affinity selection by doing the dose response, like I showed you the example. So indeed, you can do SAR, though if you've got a nice protein that behaves well in SPR, that works just as well. And we often will use orthogonal technologies if available.
Can your NativeMP platform only be assessed through a service project, or is it possible to acquire it from my in-house projects? So I think that's also a question for me now.
Yes, it is.
Yeah, so in general, of course, as said, we have several paths. How can we help on your research? The service is one of them, but we also sell our NativeMP platform as a product. So you can basically buy a solubilization screening kit and try out different conditions. And that's what we encourage people to do, is to invest the time into the screening phase, so to find the best copolymer that suits your system, where you get a good yield, where you get a good quality of the protein, a good particle distribution, and the sizes of the particles. So put a bit effort into the screen, and you will have a good and high quality protein preparation in the end.
That was
What
for me.
cycle do you use?
Yeah, so we typically use a poly-LC, polyhydroxyethyl A column. It's a 5 centimeter column, 3 micron beads, 60 angstrom pores. I did talk about our macrocycle library. Those are like 850 molecular mass average compounds. For that, we actually move up to 200 angstrom column, because a lot of them break through the 60 angstrom. But this is the same column that's pretty well described in the Merck literature, as well as some of our publications. Contact us. I'm happy to provide you with the exact specifications.
Thank you. Excision force has 6 molar affinity to isolated ACD of GLP1R. You report similar affinity to your purified receptor, as the ligand also interacts with the 7 transmembrane part. Would you not expect higher affinity for a fellow receptor?
Got me on that one.
That's a very good question. Yeah, I mean, something you will probably need to test out, right?
Yeah. How about we put this one on hold, Neil, and we'll ask some of our experts and get back to you. I honestly don't know. I don't know enough about the chemistry, the biology here to give a good answer.
Yeah, I mean, from the answer, of course, yes. So that's for sure. I mean, we use measuring MST in this moment. So what the data that I showed you, yes. So that's also a solution-based method. So I mean, we detect whenever something interacts with something. Yeah. So and this is the point here. Yeah. So but for a bit more detailed answer, yeah. So we'll make sure to provide it afterwards. Very informative presentation. Thank you very much. What will the hit rate for the primary skin be if without nanodisc control? Just want to have an idea of false positive rate.
At least several percent, right? 3, 4, 5% are pretty common for these kind of proteins. Essentially, limited soluble compounds will bind to the lipids quite potently. So much so that if you weren't to do the background subtraction with a blank nanodisc, the follow-up work to find selective binders would be way more work than actually the screen itself. So generally, you have to have the blank control, some form of blank control. Otherwise, this is going to be a lot of work at the back end.
Is there a case where you cannot find a suitable nanodisc for a membrane protein, or is it expected that for sure you will find a good nanodisc from your library? Yes, actually, you can state this, yeah. So in the end, because I mean, for sure, you have 32 different synthetic copolymers, so this is already a high probability of finding the right one. But if some troubles are encountered, yeah, so there is another way also to perform the screen, yeah, so where you can not only use different synthetic copolymers, but also vary the conditions that you screen. And what I mean about these conditions, about the temperature of solubilization, for example, about the duration of your solubilization, about the source of your solubilization, because you can work, classically by extracting the membrane pellet and then solubilizing the membrane pellet, but you can go directly on solubilizing the cell, because synthetic copolymers, at least mammalian and insect cell cultures, you can directly lyse the cells or lyse the cells by the different method, yes, and then work with the 9K supernatant. So there are different ways how to improve your solubilization in this moment, yeah, but it really depends on the target protein. But till now, yes, we were able to solubilize basically every membrane protein we had in our hands. Have you used ACD in the counter screen?
We have not. We've done some counter screening for various projects, but not this particular one. Oftentimes it's SPR or usually a functional assay, sometimes a phenotypic assay that we do downstream. We have not done ACD in this case.
Yeah. So only the empty nanodisc was used as a counter screen in this case. Hi John, sub-CMC detergent for reasonable heat rate in ASMS. Do you find 10 times below CMC and 100 times below CMC make a difference of ionization suppression is not significant?
So ionization suppression isn't really a huge problem because well, I guess it could be, but generally we usually use higher detergent than that. Our default is 0.05% between 20, which should be 4 to 5-fold below the CMC. We do want to have a safety measure there, so we don't have temperature, ion extract, et cetera. It doesn't accidentally create micelles and screw things up. But yeah, generally speaking, for mass spec, less detergent is better. A lot of the detergent is actually filtered out in the SEC column anyway. But in the first bit of the reverse phase goes to waste. We don't really see mass spec signal differences with or without detergent, but then again, we're using usually quite low amounts.
Great presentation. Thank you very much. Are you adding positive compound control in your pools? As a control, would it be not better to have another class of AB GPCR reconstituted in copolymer rather than empty copolymer nanodisk?
So the answer is yes. In fact, that's a project we're working on right now. So instead of essentially wasting the time running a blank screen, if we could run a second target and then subtract them from each other, that would be more efficient. The important thing there is to make sure that the hit rates are more or less similar and that we get similar results. So and in fact, that's what we're doing next in our collaboration. Hopefully next time with this webinar, we'll be able to give you an example where we've done that exactly. It was a little bit faster to go with blank nanodiscs in this particular case. So great observation, great thought, and that's exactly what we're doing. Having a positive control compound is always fantastic. If we have one, we can use it. In this case, we didn't. We can either add that to select pools as a 251st compound and fish it back out again. We can also run it in singleton just to make sure the system's working well. So if you have one, if we had a positive dual compound, we will absolutely incorporate it to screens.
So you heard, stay tuned. Yeah, so part 2 of the Momentum and Cube Biotech presentation gonna happen in the future. Okay, next question, please. Which kind of copolymer did you use for ASMS of GLP1R? Do you need and add any detergent and incubation buffer for these nanodisc proteins or not at all? So as I said, we perform a kind of the screening, so to stabilize a certain membrane protein target, and usually also it's worth to try a couple of polymers, so a couple of preparations to see if one is working good. But in general, once perform the screen and you see that sample behaving biophysically and biochemically stable and looks good, so that's already a good hint to work and to use it in for example, ASMS. So in our preparations, we don't need any detergent. Maybe the second part of the question.
We didn't add any either. Nope. In this case, there was no detergent added.
Yeah, but if the question already goes in this direction, so for example, if you would use some method, so where you would need a decent amount of the, for example, twin or something like this, so that's something that the system gonna tolerate, but not for a long time. So usually in these cases, we tell our customers to use the buffer that they with the detergent, so and directly perform the measurements and not incubate the protein for a long time in such a buffer. How do you check if the target is stable for duration of the screen?
So that's a good question. In this case, we knew we had a fairly stable protein because Cube had already tested that. But sometimes, I mean, it can take up to about 18 hours to screen a 50K subset. And if your protein is not stable for that time, it's a problem. Typically, you'd see that in your hit distribution, right? If your protein is unstable, you'd see hits drop off across the screen. Alternatively, you might see hit rates go up dramatically if you have a protein unfolding and now you got a disordered protein and non-specific binding happening. So as long as you have that kind of well-distributed hit rate, it's a good sign that your protein's stable. This is something we can test, right? If especially with a tool compound, we can run an experiment across multiple hours and see if we're getting similar binding affinities, and if that binding affinity starts dropping off after time. In the case where the protein isn't stable for 18 hours, we'd run the screen in tranches, right? We run the first 4 rows, throw in an aliquot, add it to the next 4 rows, so we can actually keep the protein on the deck for as little as necessary. Obviously, it's more efficient if we can run the whole thing in one overnight experiment, but if we have to run it in 2 or 3 or 4 tranches, that's fine. It's something we test and make sure is stable before we get started.
Yeah. And also from our side, yes, as a part always of the project, yes, so we test the proteins and their stability and on the freeze-thaw cycles, so that we can really ensure that the protein is stable. And what I can tell from our projects and our experience with the NativeMP stabilized membrane proteins, that they are way much stable than counterparts stabilized in the detergent, because the clue is in the lipids. Yeah, the lipids are not removed in our preparations, and they're kind of contributing to the stability. They're shielding the protein and stabilizing it, so that allows you to perform, for example, measurements over not only over hours but also sometimes over days. So it depends on the target, of course, in the end, but that's something also that can be tested in the project.
That it? More questions?
I think there are more questions. We have many questions. Yeah. Any experiments with GPCR in finding agonist antagonist using the ASMS method?
So typically we're looking for binders, right? So those experiments of agonists or antagonists would have to happen in downstream experiments. So our proteomics assays, especially our signaling pathways, could be phosphorylation channels, et cetera, are things that we can do. So we've definitely done those kind of experiments. Obviously, has been this kind of work for years. We haven't actually done a case study where we found targets in an ASMS screen that we can talk about that we've then taken to downstream experiments. But some of our clients are certainly working on asking such questions with our two groups. And so the answer is we absolutely can ask these questions and run studies like this. But in this particular example, we have not yet done that with any of the compounds we identified in the screen.
I mean, that would also require some further methods. Yes,
sure. It'd be proteomics experiments, cell-based experiments, etc.
G protein recruitments or so on. Yeah, so that
exactly.
to find out it's agonist or antagonist.
Yeah. So the answer is we absolutely can have discussions around such experiments, but in this particular case, we did not follow these compounds up.
Yeah. Okay. How long will you keep the complex at 4 degrees after incubation during the screen? Are they stable overnight according to your experience?
I think we kind of answered this question a bit already, but a 50K subset, 200 injections takes about 18 hours. So it's an overnight run. You put it on, you get it ready, then by the next morning it's typically done. And yes, most proteins are stable for 18 hours, and the plate nest is actually at 4 degrees, so it is chilled. But some proteins are not stable for that long, and we have to run the screen in tranches. That is just, it is what it is.
Yeah. Also thinking about NativeMP system, yeah. So we also encourage our customers not to forget that they are working also not only with proteins, but with protein lipid particles, and think about the physical-chemical properties of the lipids, yeah, at lower temperatures, because they're gonna be like more solid, yeah. So in this moment, yeah. So and even sometimes beneficial, yeah. So to have not 4 degrees, but even a higher temperature in this moment, so it will be at room temperature or even at body temperature, because this may also impact the membrane protein in the end. So and for sure they are stable also for this time now. Okay, that's it. I mean, there were plenty of questions. Yeah, so thank you very much. Yeah, so yes, John, thank you very much for being here.
Hosting me. Appreciate it.
And for this very nice presentation. I really enjoyed this. I already see. Thank you for this informative webinar. So thank you all for being here. And yes, we have also our other webinar series. So I just wanted to highlight the next one. So many of you probably already saw first 2 episodes together with NanoTemper. The next one gonna happen on July 1st. The topic gonna be how you can achieve highly sensitive and precise binding affinity measurements without disrupting protein conformation. So thank you very much for being here today. If there are any more questions, we're always happy to answer them if you submit them via email or in comment on LinkedIn. And yes, we'll see you next time.
Thank you very much.
Bye-bye.
Bye-bye.